Hi, today I'd like to introduce our research unraveling rodent brain signals during euthanasia with data-driven method era. In the introduction section, the project uses a data-driven approach to discover models of complicated nonlinear systems for predictions. The focus is on finding simple governing equations that accurately describe the data using appropriate coordinate transformations. ERA is useful to simulate changes in targeted rat brain signals before and after euthanasia with CO2. BEAST is a powerful tool for analyzing time series data and can provide valuable insight into the underlying processes that generate the data. Our objectives: use ERA to reconstruct the signals without prior knowledge of the governing equations. Contrast the complexity of brain signals between rats that are anesthetized and awake. Investigate brain activity during euthanasia. In the method, we consider the following system with the following impulse function response, with output measurements yk. The most important part of the method is to construct the Hankel matrix H. The Hankel matrix uses a specific number of stacks. This is important because the number of stacks is equivalent to the signal's complexity, and the more complicated the signals are, the larger the Hankel matrix needs to be. And by constructing the Hankel matrix, we can produce a lower dimensional matrices AR, BR, and CR that explain our model. It's worth mentioning some challenges we faced while applying ERA to our dataset. Applying ERA directly to the data leads to an unresponsive MATLAB notice due to the huge number of time points. To mitigate the issue, the time was divided into smaller intervals of 5 minutes. However, even with the approach, running a small chunk took 3 days and resulted in an out-of-memory MATLAB notice. The solution was to use the BEAST algorithm along with the interpolation to detect change points in the dataset. The algorithm aims to simulate the original data with fewer points while detecting all change of points. The best time discretization is found by aligning all detected points. The interpolation is used to unify and reduce the detected points with the best data representation. In the results section here, we have the experimental timeline. Then we have here 4 electrodes were implanted in the brain of each rat, a showcase of the recording for all brain channels or regions. We generate or extract the change points via BEAST for each channel, unify, align, and find the best time discretization, produces all change points, and this is made because ERA requires equally spaced data. In this figure here, we have the BEAST reconstruction via original data, and then we zoomed in to show the change points with the line segments via BEAST. In this figure, we have the ERA reconstruction and another zoom in showcase for the ERA reconstruction. In the last 2 figures, we have the relative error and the number of stacks for both rats groups, the anesthetized and the awake, showing how complex the brain activity for each group, with how good the reconstruction via ERA according to the relative error. In conclusion, we analyze the reconstructions given by the ERA method in different phases of the experiment and discuss the results for each rats group. The findings show that anesthetized brain signals are more sophisticated than the awake group before applying CO2. That is, the number of stacks for the anesthetized group is higher than for the awake in this phase. In contrast, the opposite happens when the rats are euthanized by CO2. However, compared with the anesthetized brain signals, the complexity is relatively comparable. Moreover, the ERA method approximation provides a very good reconstruction by comparing the relative error values in both groups.